Our next speaker is Federico de Angelis, and his work is about the coronavirus nucleocapsid gene that meets the eye. Thank you.
Right, okay. Hello, everybody. Hello, UK-ICN. Today, I'm going to try and convince you that there is slightly more to the coronavirus nucleocapsid gene than meets the eye. So the nucleocapsid gene encodes for the nucleocapsid protein. We've already been talking about it. It is effectively the histone of the virus. It packs the viral genome during virion formation. But the reason I find it particularly interesting is because in SARS-CoV-2, in porcine delta coronaviruses, MERS-CoV, in MHV, and other coronaviruses, the nucleocapsid gene has been found to encode in its second reading frame accessory genes. And these diagrams here represents the genes of the viruses that I've just mentioned, and all the color-coded bars that you see are effectively the genomic coordinates of each methionine codon found with these open reading frames, color-coded according to their Kozak strength, or their likelihood of initiating translation. And in our lab, we're very interested in accessory genes because they have been implied, not by me, but by other scientists, to play a role in adaptations to new hosts whenever a host jump event occurs. And so because so many coronaviruses do this thing, at the beginning of my PhD, I asked the first question of this talk and of my PhD, which is it by chance the nucleocapsid gene of coronaviruses in general is a reservoir for accessory genes? And to answer this question, I downloaded the genomic sequence of 113 different coronavirus species divided by the 4 different genera. And then I looked for operating frames in the second frame domain using an online tool called Finder, and these are parameters they used. And then I came out with a series of parameters to score the operating frames that I identified as either good, moderate, or poor accessory gene candidates. But I'm not going into details because we do not have the whole day. And this is the first batch of results. So if you look here at this graph, the number of viral species divided by alpha, beta, delta, or gamma genus, this shows the number of each species in each genus having either a good, a moderate, a poor, or no accessory gene candidate in their second reading frame event. And this graph here represents exactly the same thing, but rather as a percentage distribution, just to make sure that we have, because we do not have as many gamma coronavirus species as we have beta coronavirus species. What I hope you can immediately notice is that if you look at beta and delta coronaviruses, their N gene does seem to be a reservoir for accessory genes. Not so much in alpha, and definitely not in gamma coronaviruses. But I found this was particularly cool because this seems to be a genus-specific characteristic of coronaviruses, which implies some degrees of conservation. And looking at conservation is quite interesting because it allows me to ask the second question of this talk, which is, well, how do these operating frames then evolve among closely related viruses? And to answer this question, I did a very simple thing, and I plotted the length of these operating frames, this is the laser here on y-axis, divided by genus, and then divided by subgenus. And what I hope you can immediately notice, if you look at gamma coronaviruses, there's not a lot going on. If you look at alpha coronaviruses, there's quite a lot of variability. But if you look at beta and delta coronaviruses, at least at the subgenus level, these operating frames seem to be quite conserved, at least in length, but there are individual outliers. And individual outliers are like my favorite thing, because they allow me to ask the question whether these outliers are actually correlated with a specific type of host. So I asked the very easy question, where would the human infecting coronaviruses fall within these groups? And if you look at SARS and SARS-2, they're utterly boring. They're very average within their respective sarbecoviruses. But if you look at MERS, OC43, HKU1, and to a certain extent, even NL63, they do seem to be the relative outliers within their respective groups. And so today I'm going to tell you 2 stories, one in NL63 because we're in the Alphacoronavirus session, and one in the MERS-CoV because that's mostly my work. And so if you talk about NL63, this is the N gene, same type of diagram that I've shown you before, and it seems to have 2 major operating frames in its second reading frame event. But if you look at the other tetracoviruses, which we're going to call 9A and 9B for simplicity purposes, they're bat coronaviruses, they all have longer operating frames in their second frame event. And if you look at their genealogy using, if you look at generated phylogenetic tree using the N gene, it seems that 9A is more closely related to NL63 than it is 9B. And if you look closer at the second reading frame of these N genes, you can pinpoint out that in 9B, there seems to be this long operating frame. But then in 9A, which is closer to NL63, there was insertion of 2 stop codons, which truncated it and to make it a little bit shorter. And then if you move into NL63, actually there's another round of truncation which happened here, which truncated even further. So it seems that moving closer to humans, the operating frames in N start to get truncated. But then looking at this, we can then maybe hypothesize that the ancestral tetracovirus had an approximately 800 nucleotide long operating frame in its second reading frame that got subsequently shortened the closer we got to humans and we moved in other species. But what about mRBACoviruses? Well, we know that MERS-CoV expresses ORF8b in its second reading frame. It's an accessory gene that's been studied, there's publications on it. But if you look at all the other mRBACoviruses, they also have a longer ORF8b-like gene that's longer at its N-terminus and C-terminus. But if you look even more closely at the second reading frame of MERS-CoV, there's actually a conserved start and a conserved stop that align perfectly with the start and the stop of the other ORF8b-like genes. So much so that if you align the amino acid sequence of these proteins from the start to the very conserved start to the very conserved stop, there's actually substantial conservation even outside the core ORF8b protein, which suggests that maybe all mRBACoviruses had an approximately 700 nucleotide long accessory gene in N, but then in MERS, ORF8b actually got truncated twice for insertion of stop codons, twice. But then this is not even the full story, because it turns out that I was looking at a lot of MERS coronavirus sequences, and there's actually two different strains of MERS, one which has the canonical ORF8b, which I just shown you, and one which has a slightly longer ORF8b. So of course I wanted to know when did this happen, and I took 700 sequences of MERS-CoV and I generated a time tree. And turns out that using BEAST, and turns out there was a nucleotide mutation which emerged in around mid-2015 that elongated ORF8b, and that stayed with us until March or April 2019. And sure, there are sequences of MERS that do not have this long ORF8b, so initially I thought, well, this might be founder effect. Except that when you actually geographically distribute these sequences, turns out that all the sequences found in Saudi Arabia have now the longer ORF8b, and all the sequences that don't have it are actually found in African countries, so Kenya and Ethiopia and so on. And I thought, this is super cool. I can pinpoint the evolution of these accessory genes in a specific virus, in between different viruses. But at heart, I am a wet lab scientist. And so then I asked the question, well, does my pipeline then predict actual expression? And to answer this, I took the N gene of 6 different mRBACoviruses here, put it into mammalian expression vector, but I stuck at the 5' N, upstream of the start of N, a transcriptional regulatory sequence, and at the end of each ORF8b, I stuck a flag tag for these proteins to be detectable. Put it into mammalian expression vectors, transfected human cells, and then look for these proteins either through Western blots or confocal. And with my delight, most of these proteins are actually detectable, and they're actually expressed within their context of N. And with different efficiencies, in fact, if you look at MERS coronavirus and EGO coronavirus ORF8b, they're only visible when you treat cells with MG132, which is a known proteasomal inhibitor. So this means that the proteins some proteins are not that stable. But if I want to leave you with something from this slide, is that if you look at the short version of MERS and the long version of MERS, actually that the elongation, the added C-terminus, is making the protein more stable because now it's actually detectable. But now being able to express these proteins asks the juicy questions, right? Like, what do these proteins do? And to answer this question, I need to backtrack a little bit. I need to talk about ORF9b from SARS-CoV-2. And that's because during the pandemic, this is being widely characterized. It's been found to bind to TOM70, which targets it to the outer mitochondrial membrane, and to interfere with innate immune signaling. And of course, I wanted to recapitulate this. I had an ORF9b GFP transcript, as a construct in my library, and stained mitochondria, stained TOM70, stained for GFP, and it's bang on the money. It's perfect localization here. But then I thought, well, oh no, evolutionary speaking, mRBACoviruses here are not that far from mRBACoviruses, so could there be some signature of conservation, functional conservation there? And I did exactly the same thing, and I'm not going to show for all of these viruses, but what I hope you can immediately see if you look at Neuromycia coronavirus is that it does seem to co-localize to mitochondria. If you look at MERS, I'm sorry, it's not very visible right here, but they don't seem to co-localize to mitochondria. They form what we call in our lab these donut-shaped structures. But the staining for mitochondria is not particularly bright in the cells that are expressing ORF8b from MERS. So of course I wanted to know what was happening, and I did a Z-stack of cells expressing ORF8b from MERS. And if you look at this video, those donut-shaped structures look suspiciously like mitochondria in cells that are not expressing it, but it's just that the stain is not getting there. The mitochondria are not getting stained in the cells that are expressing this protein. And so, okay, maybe there's some data suggesting they're going to mitochondria. This is interesting. Do they also interfere with the interferon response of the cell, like ORF9b from SARS-CoV-2? And to answer this, I use a reporter-based assay, a luciferase assay, with a plasmid controlled by interferon beta promoter, and then stimulating cells with poly-IC. So once you put it inside the cells, you get light. And if my hypothesis is correct, if ORF8bs are actually dampening down the interferon response, then you shouldn't see light, or you shouldn't see as much light. And it's a bit fuzzy data. Each dot here is a biological replicate, but some of these proteins, like if you look at Neuromycia coronavirus and HKU5, do seem to be dampening down the interferon response. As a positive control, they used NS1 from flu, which is a non-interferon antagonist. Interestingly, they were stopping the interferon response more than ORF9b, but this is the Wuhan strain, and we know that the antagonism of interferon from ORF9b became stronger with the other variants. And so to finish off, I think I wanted to present our working model. So we think that once the viruses are in their reservoir, and we assume most of the reservoir are bats, they have this N gene and this platform in second reading frame, an accessory gene that probably is an interferon antagonist, or probably goes to mitochondria. We've seen with MERS, with SARS-CoV-2. But then once the virus jumps into a new host, now its N gene is not tailored to that host. And so its replication role takes preference and is prevalent compared to the antagonistic effect of the accessory gene in its second reading frame. And so the virus is going to modify its N gene, is going to tailor it to its new host, and that can lead to shortening and breakage of accessory genes in its second reading frame. But then once it establishes itself in the new host, as we've seen with MERS, then it still has that platform to have an accessory gene in its second reading frame of N that then can be reconstituted with time and then regain its role. Before leaving, though, I need to thank all these people here, in particular the Guardians of the Galaxy, and my boss here. I know we have a cool name. You should apply. And yeah, if you have any questions, send them my way.
Thank you so much. Any question? Well, I'll start with one. I've seen in the phylogenetic tree, when you look at the ORF-B, or 8B, did you look also at host? So, for example, where those MERS are isolated from, because you divided geographically.
I did look at host, but the thing is, there's so the thing with MERS, it keeps jumping on and off from camels into humans, and sometimes even from humans into camels. So there's not a clear-cut difference. So, but I see that this is not to say that's what's happening, but when you do a breakdown of human versus camel isolated cases, from the point ORF8B becomes longer, there's a slight increase in percentage of human cases versus camel cases. But this is not to say this is what's happening.
Of course.
I don't want to claim anything. It's just, you know.
Did you also find any bat?
Bat case of MERS, no, because this was mostly looking at MERS. Specifically, there's MERS-related coronaviruses, but all the MERS-related coronaviruses that I found I have other data sets that I've not shown, they all have a long ORF8B, and they're very similar to all the other mebecoviruses within that group, within the subgens. So,
anyone? Well, in this case, I guess we can go to the flash talks. And this will be basically 3 minutes, right?
Yes.
And with no question, but at the end. Thank you so much, Federico.